Namaste to everybody and pranam. We are here on the second step of KSAS talk, which we organize every month, and we are here on the second step. इंस्टीट्यूट ऑफ एडवांस साइंसेस, जो कि अमेरिका का एक प्रसिद्ध संस्थान है, उसने स्कूल ऑफ इंडिक स्टडीज के साथ मिलकर विज्ञान और मानविकी के सब के विषयों को एक साथ लाकर, अलग अलग डिसिप्लिन्स को एक साथ लाकर, जो टुगेदरनेस है, जो सहवास है, जो भारत की परंपरा है, जो हमारे मूल सिद्धांत हैं, जो मानव के मूल विचार हैं, जो मानव के मूल व्यवहार हैं, समगच्छध्वम संबद्धम समो मनासि जानता हूं, देवा भागम यथा पूर्वे सज्जना नाम उपासते, जो हमारी मूल भावना है, मानव की जो मूल भावना है, टुगेदरनेस, होलीनेस, इस प्रकार के विचार के साथ अगर हम आगे बढ़ते हैं, वेयर वी आर थिंकिंग टुगेदर विथ साइंस एंड द डिफरेंट डिसिप्लिन्स ऑफ ह्यूमैनिटीज, इस विचार के साथ आगे बढ़ते हुए INADS इंस्टीट्यूट ऑफ एडवांस साइंसेज, जो अमेरिका का प्रसिद्ध संस्थान, उसने अपने प्रकल्पों को बढ़ाने के लिए उसी क्रम में लखनऊ, उत्तर प्रदेश, भारत में कुरवोम स्कूल ऑफ एडवांस साइंसेस की एक आधारशिला रखी है। और उस आधारशिला के क्रम में हम लोग KSAS टॉक सीरीज आप लोगों के बीच में लेकर उपस्थित होते हैं और प्रसिद्ध विद्वानों को यहाँ पर हम बुलाते हैं, उनके विचारों को सुनते हैं, क्योंकि इस विचार के पीछे हमारा एक निहित मिशन एंड विजन है। वी हैव आवर ओन मिशन एंड विजन बिहाइंड इट. वी invite different dignitaries, different valuable peoples here. We have the purpose of this talk to invite experts from various fields to discuss unconventional but important perspective related to the society, religion, politics, modern and traditional science, environment, philosophy, etc. तो इसी क्रम में आज हम लोगों ने एक विज्ञान से जुड़े हुए अपने विद्वान को आमंत्रित किया है. I request Dr. Rajkumar, who is the Associate Professor in Institute of Advanced Sciences and the Director of Kurvom School of Advanced Sciences, Lucknow. I request you to please introduce today's speaker.
Thank you. Hello. I feel honored to introduce Dr. Barsegov. He was my committee member. My first meeting with him is because of Dr. Singh, because he came to University of Massachusetts to deliver a lecture, and Dr. Singh told me that you should go and listen to him. And because that time we were thinking to use some simulation for my thesis, PhD thesis, and we were looking for people who we can collaborate. So that's the same week, Dr. Valeri Talks was organized in University of Massachusetts. So I went there and I had a dinner with him. I was the only student, rest of all other were professors. So we discussed a problem which I want to address through simulation. And then we started our journey. So this was back in 2009. And since then, we build our relationships and we publish quite a few papers, very important paper related to protein dynamics. So he is very good and very established professor in simulation area. And he did some very good research. And he is one of the expert in molecular dynamics simulation. So I'm very happy that he agreed to deliver a lecture for the KSAS. So Dr. Singh, if you want to say some word, then you can also say that, Dr. Singh.
Sure, no, it's wonderful that he is able to get up that early in the morning. And I know the time difference, and it's been wonderful to work together on several projects, and we hope that we will do some more work together. He's somebody who is very open-minded, and I've learned so much from him about this simulation ideas, and hopefully we will find some ways to work together here as well. And hopefully he will visit India soon, which is what we're looking forward to. But today we are going to just listen to his talk, which is a wonderful topic here for us to understand. He may have, it's a little bit kind to explain people to audience because it's not exactly the field most people who are attending this. So with that word, I would welcome again myself also, Dr. Valeri Barsegov, and invite him to start his discussion.
Good evening, everybody. Good morning, good evening. Many thanks for providing me with this opportunity to talk about my research. I'm really honored. In my lab, my students and I, we are doing computational/theoretical studies, and we're looking at different systems. And as Raj Kumar pointed out, we have been working in the past on botulinum neurotoxin. We have quite a few papers together. We believe that that a scientific success in several fields is actually determined by a nice, very delicate balance between the simulations and theory, between the simulations and the experiment. So it's right now, if you talk to people, they take simulations as yet one more experiment that one can do. And sometimes these experiments are referred to as in silico experiments, i.e. in the computer. And so it has become very important to have theoretical or computational exploration, and it always adds up to the quality of the research. So I will talk about today. What I will talk about are the new directions that we're taking in my lab, so there's this platform that we have built, my students and I. It's called CellDynaMo, CellDynamics, but it's a very broad platform. It can be applied to study the processes beyond the processes associated with the cell. So one of the applications that I will talk about today, the main application, is mitotic spindle assembly. There's a huge paradigm in the cell biology that the chromosomes have to be segregated in the center of the cell. So you see here, I'm pointing with my mouse, and before any cell division can occur, they have to be properly aligned and segregated, and then after that, the cell division takes place. If anything goes wrong in this process, then what happens is that either the cell dies or it might also lead to pathological conditions. So that will be the first application. The second application is having to do with the blood clot. So there's this nice machinery, biochemical machinery, during which there's this polymerization of fibrin monomers to longer polymers, and then to oligomers, and then to protofibrils. Then protofibrils laterally aggregate, and what you see is that the formation of the extended three-dimensional meshwork, also related, also known as fibrin clot. Now, this clearly occurs in the blood vessels. So you have lots of different cells in the blood, and so therefore a lot of these cells, for example, here you see the platelets. These are the important cells. They also get involved. Okay, and so there are several aspects of blood clotting that are very least understood. So, for example, what is happening after the clot has been formed.
Excuse me, Dr. Barsegov, your slides are not moving.
Yeah, I know. I'm just starting from the first slide. I'm trying to explain what I'm going to do. Yeah. So and so here there are lots of unanswered questions, and we're starting to dive into these questions, and we have to have a vehicle for these things. And for us, for an experimentalist, a vehicle is experimental apparatus, and then sometimes it takes very long to build one. In the simulations, we have to have a methodology, if you will, computational methodology, and we have to have a platform or a software package, and then we can do things. And the third thing I wanted to say was, before I start moving the slides down, is that we perform all the simulations on GPUs. This is a revolutionary development in the field of computing, where people have started, to realize that not only can you play games in the computers, video games, right, but you can also do computer simulations, so do something more useful or more meaningful. So it's a short, short outline of the talk. I will talk about methodology very briefly. Then I will talk about the application to mitotic spindle assembly. Then I will mention few recent developments in this direction, and then I will point to new directions. So the computational methodology. So like I said before, we're doing simulations on the GPU. So therefore, all the algorithms have to be ported to GPU platform. So it's a hardware. So we have cluster, and so each of these nodes has 4 GPUs of various complexity. And so basically what you have is that one GPU substitutes a desktop. So therefore, you can have one graphics card like this. It's called NVIDIA A100, and if you run simulations of many, you can tackle complex biological systems. Now, in terms of the computations, the algorithms really depend on 2 things: the system size and the time that you want to span. Okay, so if you are interested in small conformational changes here in proteins are having from 100 to 1,000 particles, then you have to involve molecular dynamics simulations. Now if you want to go to larger time scales and larger systems, for example, we're interested in cells, or we're interested in virus. So this is a virus, hepatitis B virus, with the double-stranded RNA in it, and so you have here counter ions floating around. So you really have to change the representation. You have to go to coarse-grained model, or agent-based model, or reaction diffusion stochastic methodology if you're interested in modeling the entire cell. Okay, so different, the choice of methodology really depends on the problem that one wants to address. Okay, that's the message. Now, let's start with the CellDynaMo package. So we're solving numerically the so-called reaction diffusion master equation. So in the cells, what are the processes that are happening in the cell? Well, particles can diffuse. Okay, there are chemical reactions, and then there are also mechanics, right? So for example, microtubules can push or pull these chromosomes to the center or from the center towards the pole. You see here 2 poles, and then you have microtubules growing and shortening. And then you have this, as an example of 1 chromosome, okay? And there are lots of different agents working here, different enzymes, different moieties. And so you have 3 processes that you have to describe simultaneously: the chemistry, which is the reactions, right? So you can have association, dissociation reactions, enzymatic conversion reactions, etc. Then the particles, the substrates and the enzymes and the large biological particles can also diffuse. Okay, so therefore you need to have diffusion. But then when you have mechanical interaction, you have to have the mechanics. You have to be able to describe the mechanics. So therefore we have the reaction diffusion master equation for diffusion and reactions, chemical reactions, and we have Langevin dynamics. That is when the mechanical interactions are involved. Okay, and we have the platform. It's called CellDynaMo package. It's a freely available software package that enables us to study different processes. So just want to say one thing about the complexity of the system. The cell is such an object that you have lots of different orders, lots of different length scales. So you have the particles that are spanning length scale from anywhere from nanometer to micrometer. So for example, microtubules are micrometer long. This is why they're called microtubes, basically. So therefore, we're spanning 3 orders of magnitude in scale, in length, but then also 6 orders of magnitude in time. So the processes that are happening in the cell, they are of the order of microsecond to minutes. Okay, and so with this package, using sophisticated description, we can actually span these many orders of length and 6 orders of magnitude of time. I don't want to get into the details of what is doing what, but there are lots of different enzymes. So let me just focus on the most important ones. So you have the Aurora B. Aurora B performs phosphatase. of the microtubule associated proteins. So these are decreasing the lifetime of the microtubule kinetochore attachment. So what are the kinetochores? So each of these chromosomes has this little corona area to which microtubules can grow and associate. So these are these little bolts and little area, curved area that you can see here. This is a chromosome in the ball and spring representation. And so microtubules grow and push towards, in this area from both poles, okay? And so you have here these red particles, red colored particles. What they do, these are aurora B particles. What they do is they phosphorylate the microtubule associated proteins. These are these, flexible linkers that you can see in them. So you see in here. And, by phosphorylation, you are thereby decreasing the strength of the bone, okay? And then there should be a feedback. So there's another enzyme called phosphatase that's dephosphorylating these attachments, okay? Thereby increasing the lifetime, thereby increasing the strength of the bone. So you have two, opposing processes, decreasing the strength of the bone and increasing the strength of the bone, okay? These are the two most important enzymes, aurora B and phosphatase. So, and of course, we have to model them. There's a certain number of them, copy number. And then you also have aurora A. So aurora B is somewhere here. Aurora A are centered around the two poles. Now, from these poles, microtubules grow. And then as they grow, they start interacting with the kinetochores, okay? So we're talking about thousands of different enzymes, thousands of microtubules growing from both ends. And then we want to understand one important question, and that is, with seemingly chaotic dynamics of so many chromosomes and so many microtubules, how come that we have, the right speed and the right accuracy of these processes? That's basically the question. And so clearly something can go wrong, okay? So these are called errors or erroneous attachments. And then, the second important question is, how these errors being corrected as the cell approaches the time when it has to divide, okay? So this is called prometaphase, part of the cell mitosis, okay? So there's a little very short phase before the cell division occurs. It's called prometaphase. Now, some attachments are accurate. So for example, here you have two poles. Microtubules are growing from both poles, okay? And so out of 4 possible attachments, 3 are wrong or incorrect attachment. So that's monothelic attachment, when the chromosomes connected only to one pole, but are not connected to the other pole. Merathelic, that means that the microtubules attach to both sister chromosomes, okay? Sister chromatids, okay, to both kinetochores, okay? So this is also a wrong attachment. Then you also have synthetic attachment. That is when one pole attaches to two kinetochores, the other one doesn't. And then there's only one, it's called amphithelic attachment. That's when each pole is connected to just one kinetochore. So the left pole is connected to kinetochore on the left here, and then the right pole is connected to the kinetochore on the right hand side here, okay? So these are called kinetochores. These are the most important parts of the chromosomes, okay? And then of course chromosomes have arms, so we have to model those arms. So the way you approach it is you try to develop different parts separately. So for example, first we wanted to understand how microtubules grow and shorten, and luckily there are rates of microtubule growth and rates of microtubule shortening. These are all known experimentally. And then there's also frequency of catastrophes and microtubule catastrophes and rescues. So what are these is that the microtubule grows, and then at some point stochastically it switches to shortening, okay? And so there should be a frequency associated with these, with the catastrophe. And once it starts shorten, at some point it decides that it wants to stop and start regrowing again. So that's called, microtubule rescue, okay? And so there's some frequency of these events, okay? So luckily, all these have been measured experimentally, and their function of the tubulin concentration, and all these kinetic rates have been measured. And so, once we know that, and once we model them in experiment, so therefore we can have a cell model. Okay. So here you have two poles. The microtubules grow and shorten from both poles. Okay. So therefore we're good to go. So we can model these. And now this is just one element of this process, right? Microtubule grow and shorten. Then you have chromosomes. Then you have a whole bunch of other things. And so, but look at the complexity of the system. You have 1,500 microtubules. Then you have kinetic or chromosome pairs. Then you have NDC80 binding proteins. These are the microtubule associated proteins that might get phosphorylated or dephosphorylated. There are about 6,000 of them. Then you have Aurora B enzymes. There are 25,000 of these Aurora B enzymes. Phosphatase enzymes. There's almost 7,000 of these enzymes. So you have a total of 15 different chemical reactions in addition to diffusion. Okay, so it's a very complex system. So, and then we need to test the diffusion. Okay, so luckily, Mr. Einstein derived this equation, which gives you the probability of diffusion of a particle at some time t in the future away from the initial position x0, and that's given by the Gaussian distribution here. So we can model that. So what you see here is a comparison of the results of simulations with the exact result with the formula. So these are the dashed lines compared with the solid lines. And what you see is that they agree fairly well for all time instances, starting from 1 second to 20 seconds. Okay, and this also shows the spreading of the spreading of the packet. And then we also need to model kinetics. Okay, we need to understand. So we need to, and we, here what we're doing is we're trying to figure out whether we have the proper description. This is a benchmark testing, so to say. And from the chemistry textbook, you have the two-step kinetics, irreversible two-step kinetics. A goes to B, B goes to C with two kinetic rates. And then you can also have reversible kinetics. A goes to B, B goes to A. So these are given by these exact equations for the probabilities or populations of particles in state 1, A, B, and C for this kinetics, and then A or B for the two-step reversible kinetics. So what you see here in the movie is is A goes to B, B goes to C scheme. So red are the A, then you have blue are the B, and green are the C's. So what you see is that initially you had a lot of reds, then you saw some blue, but now everything converts to green, right? So therefore, we have A goes to B goes to C reaction, and it's described very well. And these are the simulated curves, and they're compared with the theoretical curves that are given by this equation. So we're good to go. We know how to model diffusion. We model correctly the microtubule growth and shortening, and we're also able to model the kinetics. So now we're applying all this methodology to describe the cell mitosis. So I'm going to go quickly over these slides because they're very technical, and it's very research specific. So you may or may not be aware of certain things. Let me just focus on the most important things. So there's a thing called bare chromosome. That is when you have kinetochores without any chromosome arms. Do you want to study these first because you want to understand what determines the orientation of these chromosomes in the cell? And there are things that we can measure in the simulations. For example, the angle. Okay, this angle, how it's oriented towards these two poles. And then we also have, we can also measure the distance between the chromosome and the pole, one or the other distance in here. So what you see here is that this is the bare chromosome, the corona area where the microtubules attach themselves to the chromosomes are here. And there are lots of these red particles, which are Aurora B particles, which perform phosphorylation reaction, thereby decreasing the strength of microtubule kinetochore attachments. And we can look at the statistics. We can see, and we can look and say, okay, these attachments are meritelic, these attachments are monotelic, these are syntelic, and these are the correct amphitelic attachments. And so therefore we can, we can basically we can basically test what elements and what features of the model, what properties, for example, lead to large number of proper attachments and decrease a number of incorrect or improper attachments. Okay, so here's a movie that microtubules grow, and then you have a kinetochore, two kinetochores, and this is the bare chromosome. So these are two coronal areas to which chromosomes, if they grow, they start interacting and then become attached. And once they become attached, remember the catastrophe, the microtubules start shortening. So therefore, there's a competition between the two poles for pulling a chromosome to one of these poles, okay? But the chromosome has to be somewhere in the middle, okay? So this competition somehow, should be balanced in such a way that chromosome ends up being in the center, as you saw in this movie. So it's called beneficial geometric feedback. This is something that we have discovered. Once you have correct attachments, then that improves the angle. And so it better orientates the chromosome, with regards to these two poles. And then it improves the attachment accuracy. And then there's another thing which is called geometric vicious cycle, which you can see in here, is that when, more attachments are formed with one of the poles than with the other, and then as a result, the chromosome is being pulled towards one of the poles, okay? So these are improper attachments, and we want to understand the mechanism, how it happens, okay? And now we understand that, just bare chromosomes are not enough to have proper attachments, because by just considering these gives rise to the so-called geometric vicious cycle. So more attachments with one of the poles results in the chromosome being pulled towards these poles. These are also known as poleward wind, and they have been shown experimentally. Now, so therefore, what we need to do is we need to improve the model. We need to add new elements. And so here, for example, instead of bare chromosomes, we're now putting chromosome arms. And back in the 2 years ago, we only had the bead spring representation, which we have been using in the first paper. And what you see here is that once you put chromosome arms, the kinetochores are not being pulled quickly towards the pole, because there's a viscous drag on these arms. Because remember, the whole thing is happening within the cell. So we have diffusion, so we have proper viscosity. And now the fact that you have these bulky chromosome arms, this prevents, the erroneous attachments. And what you see is that you have an increased number of proper amphithelic attachments, okay? So the next element was, we want to understand how the centromere, this is the, you can think of the softness of this particle, there's actually, it acts as if you have a spring connecting the two particles. We want to understand how softness of this element of the chromosome structure affects the accuracy. And it turns out that there should be a balance. So, there should be a balance in the softness of the centromere. So what you see here are a few snapshots from the simulations where the microtubules grow, and through NDC80, these are the microtubule associated proteins that attach to the surface of chromosome. These are the attachments that are being formed. And so, you can have a situation where it's very soft, or you can have a situation where this corona area, which is the kinetochore itself, is stiff, okay? So we can model the stiffness of this area. Remember, these are all proteins, so they have to be soft. So there should be a balance between the stiffness of the centromere region and the softness of the centromere region, okay? And so that can also be tested using our software package. Next, so right about 1 year ago, we started implementing the so-called cylinder potential. So now we have cylinders. So instead of spheres, now we have cylinders. And the reason why we need that is because cylinder representation of the particles in the cell enables one to simulate the excluded volume interactions, okay? And secondly, we wanted to start adding more and more elements. And so these are the chromokinesins. These chromokinesins, they live on the surface of chromosome arms, but once the microtubule comes close, these chromokinesins, they attach themselves to the chromosomes and start walking on them. And as a result, you have better description of the mitotic spindle assembly process. And what you see here is that if you just look at the movie, it looks almost natural, right? So this is the cell. These are the two poles, microtubules. You have all these enzymes. you have microtubules growing and shortening, but now we have, the chromokinesin motors. There are, a lot of molecular motors that exist in the cell, and chromokinesins, they facilitate the coupling between the growing and shortening microtubules and chromosomes, and they, like I said before, they dwell on the chromosome arms. We also found out that it really depends on the initial configuration. So you have in here chromosome, and configuration 1, it's symmetric in the center, and the 2 kinetochores are pointing towards the poles. This is the most favorable orientation that results to proper attachments. But then there are also orientations that are not resulting in proper attachment, and that is when the chromosome is coming closer to one of the poles initially, and then the kinetochores are away from the poles. Okay, so for example, configuration 5 gives rise to a large number of incorrect attachments. And the second element is that we know that there are up to 20 chromosomes that can be in different cells. There could be 8 or it could be 20 different chromosomes that have to be properly aligned in the center of the cell. And it turns out that, once you have the crowding, due to the fact that you have not just 1, but 20 or 10, many chromosomes, the accuracy drops because of the crowding. So these are the things that, these are the facts that we found from the simulations. In addition, now we're adding, so now that we're adding chromokinesin to the system, okay, and so, biologists have measured the force-velocity relationship. Apparently, chromokinesin can be stopped once the pulling force exceeds 50 piconewton. Then these motors stop walking on the surface. So there's a balance between walking and mechanics in the cell. And so what you see here is that this is without the chromokinesin simulation, and you can see that there are a large number of proper attachments. And then these are the simulations with chromokinesin, and what you see is that this is as if somebody blows from the pole. So the chromosome comes close to the center, away from the pole. These are called anti-poleward winds, and it looks like the addition of chromokinesin motor, which is a part of the system, but we're now adding it little, one by one, because we want to build, from bottom to top the system. It starts, it keeps adding and it keeps improving the statistics of proper attachment. Okay, so recent developments. So what you see here is an experimental snapshot of the mitotic spindle. So what happens is that these microtubules, they aggregate to form fibers, okay? And so there's an enzyme called kinesin 14. It lives on the surface of microtubules, so it's attached to one microtubule, and then it walks on the surface of the other one once the other one comes close. So you see in here. So therefore, there's so these two microtubules, they start bundling together because kinesin 14, binds them together. And there was an open question, in biology. The cell biologists, they don't know what's the dissociation rate. The association rate was measured, but there's gap in knowledge, and that is, what's the dissociation rate? So we tested, two different extremes. Of course, we just took order of magnitude, 0.01 inverse second or 1 inverse second. So for 0.01 inverse second, the bond is very strong. Okay, so nothing happens. There are no changes in the, suppose that there's an error. There's no mechanism for the error correction here, okay? And it doesn't look natural here. However, if the dissociation rate is 1 inverse second, then you have proper bundling. And imagine that you can have many of these now start interacting, microtubules. And so these are in the presence of kinesin 14. These are, this is the molecular motor, kinesin 14. It has a tail with which it's attached to one microtubule, and it has legs. And it literally walks on the surface of the other microtubule, okay, with this velocity, which is the force-dependent velocity, okay, 1 micrometer per second if there's no force. But once the force is pulling the microtubule, then it might stop when the force exceeds 50 piconewtons, okay. So these are the new elements we're adding to the system, and another element is dynein. That's another molecular motor that sits on the surface of so-called short microtubules. There are these short microtubules that grow on the surface of kinetochores. Okay, and again, so once these short microtubules interact with long microtubules, so these long microtubules grow and start interacting with the short microtubules, and the element that couples them together is called dynein motor. And again, it's a motorized particle. It has, force velocity relationship. It's similar. All these molecular motors, they have similar force velocity relationship. And but again, there was an open question as to what the dissociation rate constant. We tested two values, and it turns out that 10 to minus 2 inverse second is the right value. Somehow this movie doesn't work, the second one. Anyways, now we have, and the third one doesn't work. Hello?
Yeah, we are here.
Yeah.
Something happened to the, I cannot move anything.
I think your on rate has slowed down.
Yeah.
Yeah, you shared again. It's probably done.
It's okay now, I think.
Stop.
Now it's separated, it looks like it.
Okay, so let me reload this again. So now we have these short microtubules and long microtubules. And again, we have dynein, we have kinesin, we have all three types of microtubules, okay? And so, we're trying to model this. We're adding piece by piece the elements, to make the system as complex as it is in real life in the cell, okay? So, so these are the new elements. Now, the new direction. So, my lab is also heavily involved in the direction called hemostasis, biomechanics of hemostasis and thrombosis. So we want to understand the mechanical properties of blood clots, what is happening after it has been formed. And so, here's the movie of blood clot contraction. So you have fibers. These are red fibers that you see here in the movie. And you have platelets. This is the bright yellow-green particle that you can see here in the middle. What you see is that experimentalists are now capable of making these movies. These are very small objects, right? And so you see here, this is one platelet, but then you have other platelets floating around. And what happens is that these platelets actually pull on fibrin fibers together, and then that results in the clot contraction. So you have to have proper clot contraction, and after that, there's a process called fibrinolysis. That is, clot has been created, but once it has performed its function, it has to be properly lysed. Okay, so it has to be of the right structure, and the enzymes have to be able to float around and attach to different fibers here and there, cleave these fibers, and then basically dissolve, just like in this movie here, dissolve the entire fiber network. Okay, so you see in here, that's the fibrinolysis in vitro fibrinolysis. So there are lots of different questions that are not answered at the moment, and so we are working with experimental lab from UPenn Medical School on different aspects of this system. What you see here is yet another application of CellDynaMo. So CellDynaMo is a platform. So it's a set of methods and a certain collection of algorithms that enables you to tackle sophisticated problems. So we always enter the game when sufficient number of experimental data has been gathered, so that there will be very little of guesswork that we need to do on our behalf. So what you see here is that experimentalists have measured the properties of the fiber network. So for example, they know the total number of platelets. Now, platelets have these little arms. There could be up to 8 little arms that are growing and shortening. Once they grow sufficient length, at sufficient length, and if they see the fiber, they firmly attach to the fiber and start pulling the fiber. So the rate of growth and shortening of filopodia, platelet filopodia, is well known. The mechanics is well known. Everything has been published. So these are all the experimental quantities that are assembled here in one table. The most important thing is that we know the distribution of fibrin lengths, the distribution of fibrin diameters. Okay, we know the number of platelets, we know the number of filopodia, and all the mechanics, the bending rigidities, the stretching rigidities, the number of fibrin branch points, all of that. All of these have been measured. So it's a good, moment for a theoretician to enter the game and perhaps address some of the questions that the experimentalists cannot. Okay, so that has been the approach that we have been taking in the lab. And again, we're ready. We know now with CellDynaMo, after modeling such a large and complex system as cell, we can raise the stakes and start modeling an even larger system, and that is it's a macroscopic object. It's a blood clot or fibrin clot. Okay, but approaches are the same. So we can model using the experimental inputs. We can model the thickness and the length of fibers, and we have these platelets. What else we have? We have number of branch points. So fiber network is branching out. The fibrins are branching out, and so there's a density of 3-degree nodes and 4-degree nodes that have been measured. All of that is known. We can put it all in the model and start performing simulations, and perhaps start addressing questions that experimentalists cannot. So, this is a comparison of our simulations with the experimental kinetic curve, and that is the extent of contraction. So what you see is that with the parameters of the model that we have, and for a sufficiently large system size, the results of experiments and simulations agree very well. So here's a movie of the clot contraction. Although the movie is playing fast, it's actually spanning 30 minutes of the clot time. 30 minutes. Simulations can now describe biological processes on almost an hour time scale. Okay, and that is due to the advent of fast computers, GPUs, and the methods, Monte Carlo simulations, Landry dynamic simulations, Gillespie algorithms that we have been developing in the lab. Okay, and so here what you see is that there are different ways of measuring the success of the clot contraction. So for example, we can measure the area of the compacted fibrin fibers. So for example, here they're separate, and here somewhere they are assembled together forming bundles. And so we can measure the area of that. And so these are the simulations compared with the experiment as a function of time. Good agreement. Now the platelets, as they pull together, they also tend to aggregate to form ensembles of platelets. And there's a way to measure the area of platelet ensembles or associations. So here you have experimental data, here you have simulations. And so, we're not, very far from experiments. Of course, one should not expect that we'll have a photographic agreement. So agreement like this is state-of-the-art agreement, actually, okay? And then the platelet speed. So we have, the platelets move. So for example, this platelet stays where it was, but some of the places that are close to the center, they're moving. So they're moving from point A to point B, and there's a rate of motion. There's velocity. So this is the experimental velocity. This is the simulated velocity. Again, good agreement. And there's a thing that we did not know, and that is that when the system is not too big, the properties that is of the system are actually depending on the system size. And once the system size is big enough, then the properties do not depend on the size of the system. And in physics is called thermodynamic limit. Okay, and so here we have the results that show that only for systems this large, we're almost getting to the regime where the properties of the system do not depend. So you see this, this levels off, forms a plateau, the properties, and in this particular case, it's the slope of the kinetic curve. It stops depending on the system size. Okay, so we can look at the small system size, larger. This is the largest. At this point, this slope stops changing. So therefore, we have reached the thermodynamic limit. This is something that we did not know before, and so it took us about 6 months just to realize this simple fact. So there are a lot of unknowns in this murky water, but we're trying to clarify things as we move along. And the last thing is the clot rupture model. So it's important to understand, how the clot disintegrates, and it has to disintegrate normally. So if you have rupture, premature rupture due to blood flow, certain part of the clot called thrombus, it can detach from the clot, and then it can dislodge into smaller blood vessels, and that might result in the conditions associated with heart attack and stroke. And so it's very important to understand how the clot ruptures. And so these are the experiments, where what we do, and these are the experimentalists from a collaborating lab at UPenn. So what they do, they make a small incision in the fibrin network, in the fibrin gel. So this is the fibrin gel, plasma gel, no blood cells. And so once you make a small cut, then you start applying force, stretching the clot, and then at some point the clot stretches. So there's a stress strain curve that you can measure, and then at some point clot ruptures, and that results in a sudden drop in the stress as a function of strain. Okay, and so it's important to understand how it ruptures, and we now are working on the structure of the entire fibrin fiber, and once we understand that, we'll perform simulations. But before that, we can also make a model of just fiber network without any platelets. So it's just plasma rich sample. And so again, we have these many model parameters. They represent certain features of the system, real system. We have simulations, and these are the experimental stress strain curves. And so these are these dashed lines, and these are our simulated stress strain curves. The important thing is that this is the movie. We clamp one end and then we pull the other end. And so in the college notation here is that blue are the ones with low energy and the bright are the ones with high energy. Okay, so we can profile the distribution of energies, and we can also profile the distribution of stress and distribution of strain. We can understand the mechanism of rupture. What rupture first? Short fibers, long fibers, thick fibers, thin fibers. A lot of questions can be answered. Okay, so with that, I would like to stop. I don't think that there's anything else. I would like to thank my collaborators on various projects, and I also would like to acknowledge the support from NIH and SF that I have had over the years, and I would like to thank you for your attention. Many thanks.
Thank you, Dr. Valeri, for this wonderful talk and wonderful presentation. And beautifully you explained the CellDynaMo and the works in your lab. But we have here some social scientists, our students from humanities, so it is little hard to understand, to grasp completely this scientific talk. But we are very fortunate that we have a bridge. जी हाँ, हमारे पास एक सेतु, वो सेतु जो हमें समाज विज्ञान को या हमारे मानविकी के चीजों को वैज्ञानिक दृष्टि देता है, और वो विज्ञान के चीजों का सामाजिक अनुप्रयोग, the social application for scientific research. We have a bridge, we have our own bridge, and we are very fortunate to have such a bridge. The bridge, हम लोगों के जो ब्रिज हैं, हम लोगों के सेतु हैं, हम INADS के डायरेक्टर, KSAS के डायरेक्टर, प्रेसिडेंट प्रोफेसर बलराम सिंह सर हम लोगों के बीच में हैं, जो हम लोगों के लिए इस कठिन कार्य को सेतु के रूप में आसान बनाते हैं. So I want to invite Professor Balram Singh sir to comment on Professor Valeri's talk. So over to you.
Okay. Well, it is difficult for almost anybody to understand this kind of presentations. It may be even Professor Barsegov also difficult to understand this level of complexity and its relevance, especially when he's talking about biological processes which are happening at such a speed, variable speed, that it becomes very difficult for him to even defend everything that he just said. It's easy to do, like he said, it is done in silico, which is much easier to do, believe it or not, no matter how complicated it is, than to really do it in reality. That said, I think it's wonderful to have an insight in the process that is so, a couple of processes that he talked about. that are so relevant. You know, the cell division is the basis of our life and our existence. So how does that happen? Well, there are, you know, it says cell division in which you have chromosomes, they have to duplicate, and then they have to separate so that they become different cells. This process is known for a very, very long time. But the level of the speed at which and the time scale at which these things happen are not even possible to do with current technology in molecular biology. So what he and the people of that expertise are trying to do is to give an insight as to what is going on in the cell at the level that may be relevant. I think for us, you know, we can't really think more than less than in millisecond, even if we think that our brain works. But here is something that the processes are happening at very, very short time scale. But he has used some experimental data that are happening at seconds and more than seconds level. And he has to do these simulations for minutes and perhaps longer than that. Because at very, very low time scale, we have done some work with Dr. Rajkumar on molecular dynamics, where a molecule, here he's looking at the whole cell, and which is in at least in terms of sizes would be, I would say it is in microns at least, and we are talking about molecules which are in terms of nanometers at the most. So we are talking about at least 1,000-fold difference. That when he tries to do that, it becomes very complicated. Now, Alok is telling me to find a way to translate it to people who are in the humanities. While humanities people have actually better ideas in some of them, because they can actually make things happen instantly. So because they don't need to really prove anything, they can just say that this is what is happening in the entire world. This is happening at one moment. So they do have actually a sense of time, which is what I think is important. Now, from the humanities, philosophical point of view, even scientific point of view, this whole game is that of time and space. The whole thing that he is talking about, really time and space, how the time plays a role in making things happen and making things move in space. And that's what all the thing is. In this world that we exist today, it is the only question of time and space. The rest of the world, which is, I think, from, if I remember correctly, from somewhere is 96.5% or something, is dark matter and dark energy, where we don't even have time and space. So he's describing what may be known as in Sanskrit Jagat, you know, the world that we see today is humongous. And then he brings it down to our, you know, we have about 40 trillion cells. He's talking about how those at the cell level, how are things happening? How are these things happening? And there are millions of things. I remember talking to him once at the level that molecular dynamics works. If we had to do the simulation at that level, where he's talking about some enzymes, he's talking about certain elements in this, whether it's phosphorylation or some other enzymes, Aurora enzyme, how that enzyme is working to facilitate this process of assembly and disassembly. If you had to do it at the femtoseconds or nanoseconds, then it will not be possible. From what I remember, he told me once about a year ago that even one cell, which exactly what I was asking him, can we simulate one cell operation? If you had to go to that level of time, it is not possible with all the computing capacity that humans have developed so far. It's not even possible to do that. And God knows whether the real processes happen even faster than that. Actually, in Indian context, Indian philosophical context, the things happen much, much faster level than here. So I don't even know. I don't know. Maybe I will be one of the questions I would like to ask him is what is the time scale that matter moves? Matter, I mean the nuclear, subnuclear particles, and if we had any context of that, whether they affect this what is so-called biological system, because we are biological based on this material world that he just described, cells are moving, this and but why do they move? Why do they have to divide? Why do they have to reproduce? That would be one of the questions one could ask. But for our friends who are in humanities and are interested in this, and I'm glad that they stayed for the seminar, for whole seminar to listen to. That means they are very curious about it, and this is really a fantastic seminar from that point of view. If you just forget about all the equations that he was talking about, all the enzymes that he's talking about, it's just a phenomenon happening inside one of the 40 trillion cells that we have. And and that too is a certain scale that he's trying only certain aspect of it, explaining how that may be happening. Especially, I think maybe more interesting, maybe the blood clotting that he's talking about. I would even ask again, my second question would be, this blood clotting that he described, is this universal, or is this depends on where in our body is, especially the kinetics of it? And it may be actually relevant to something like that happens in a stroke, because the blood clotting in many cases, blood clotting is the initiation of those strokes, and whether there is something that can be distinguished one from the other. But going back to the blood clotting and the process that is involved in, biologically is an intricate process that may be more relevant, especially I think he talked about how to maybe find a way to declot or disintegrate the clot, how that thing can be moved. And there may be some biological systems or herbal systems or exercises like, you do yoga or other exercises, and whether that has an effect on blood clotting, either formation or dissolution of the clots. So those may be relevant to the people who are our friends in humanities. And this is a visual that you cannot see, because some of these things that he's showing, it's not possible to do biologically. So what the scientists like him, they do, they understand individual forces, and then they simulate those forces. And stochastic means randomly, it is not something that is, and this may be a contradiction to some of the humanities people who may be thinking that, our body is made by certain forces, sometimes people call them gods, and then everything is designed to do in the way it is supposed to be. But if you pay attention to his terminologies, that scientists do not believe in that. Scientists believe in that everything happens randomly on its own, and those are the things that are determined by the forces, the individual atomic forces, molecular forces, cellular forces, and catalyst. Enzyme is a catalyst, and the catalyst works in certain ways. I think someday in the future, and that's probably going to be my last thing that I want to say, because I don't want to take so much of your time, and maybe some people have questions, including myself, that maybe someday with these iterations of silica-based simulations, as well as more biological measurements and more thinking from the state of mind that supposedly philosophers may have, or the yoga practitioners may have, that they can perceive these things differently, and maybe they will be intermixing, and that KSAS has. We have other linguists, and we have, of course, scientists, experimental scientists, that they may be able to join hands together, ask some interesting questions, and expect each side to be addressing the questions of science from philosophy point of view, and philosophy from science point of view, and the expression of those information. This is also a form of expression that he's showing all these visuals so that you can just see it. other people articulated words, and hopefully all that will come together, and we will find better answers and better questions to ask, and so that we can advance the knowledge in the future.
Thank you very much, sir. And now it's quite easy to access it.
So, Valeri,
But
is that somebody asking question or just talking to themselves? Okay, Valeri, you want to answer my question?
yeah. Dr. Valeri, please.
Yes, Balram, you asked, 1 million question, $1 million worth question.
Well,
You know,
send me
life
the
is
check. Please send me just $100,000 check. That would be good enough.
I'm sure that the answer to your questions exist, but we're not, there yet to address these questions. And, maybe another, so many years of development and, thinking, intellectual labor, etc., might, bring us to a point when we'll be able to address these questions. And, yeah, life is a complex phenomenon, and things are happening on different time scales, different length scales. There are different laws of physics that are working on different length scales and time scales. If you talk about nuclei and electrons, we talk about Schrodinger equation. This is quantum physics or quantum chemistry, depending on the application. If you go to, molecular transport, then we have Fick's law, then we have the laws of diffusion. So these are entirely different, mathematical formulations, that are working at that level. And so, each time scale and length scale brings about different methodology, different laws of physics, different descriptions, different set of ideas, different set of assumptions, and different logic, actually. And, I'm sure that at some point, there's a bridge that can, link the two world, if you will, and this is the quantitative science and qualitative science. So the law, the world of philosophy, and the world of biophysics, for example, or the world of natural sciences have to be connected at some point, somehow. Okay. And of course, if you look at the early philosophers, they were, like Archimedes, for example. He was a philosopher, he was also a geometer, and he was also helping Carthage to defeat the Romans. So he was building all these, fortifications, etc. But as we progressed, then we become, because the science is so complex now that you cannot be like Isaac Newton, who worked in mathematics, in physics, and also in philosophy. So we have to choose our field, and and to be successful, we have to be focused on very small area of knowledge, and it's very hard. And I guess it takes a brain of a much larger person than I am in terms of thinking, in terms of mental capacities, to connect the two worlds, if you will. But I'm sure that that there's a way to connect the two worlds, and there have been multiple attempts, to bridge the gap between the two ways of understanding life at the macroscopic level and the microscopic level, if you will. And the answers to all of your questions, they do exist, but we have to find them. We have to find these answers.
I have just one follow-up, because I think you said wonderful thing that people need to cooperate. I wonder, Valeri, if it is possible to maybe a more phenomenal aspect of life. Okay, is it possible to simulate dreams?
Well, it's I'm sure that it's possible, but one has to do the dynamics of the neural network that is sitting inside the brain, and nobody understands what's happening when we're sleeping. You're absolutely right. One thing is that there's this blood barrier that is being surpassed when we're sleeping, and that's when the brain starts relaxing, and then there's there are metabolical processes that are happening. But this is all biological, just pure biology. And the dreams, daydreams, night dreams, but in order to understand them and maybe model perhaps at some point, we have to really understand how the brain works. And so there's ongoing research. I'm not in that field. I'm not familiar with that, but I'm sure that at some point in the future we will understand the mechanisms of memory, short-term memory, long-term memory, and things that we tend to remember. There should be some biophysical or physical, mechanism or biochemical mechanism, whatever you call it, that can, that account for these phenomena. These are phenomena that occurs on a large length scale. The brain is a macroscopic object. It's very sophisticated. There are lots of different neurons that are firing, that are connected, that are disconnected. Some are connected, some are not connected. And I'm sure it's going to be a function of the connections. It's going to be a function of the speed with which they fire, and certain things. And there's some things that are happening without us kind of participating, and that is that's when we're dreaming during the night, being completely relaxed, or during the day when we're free from doing our everyday's activities, we start dreaming. But I believe that these processes are entirely possible. Perhaps somebody out there is already simulating them, and I won't be surprised if somebody has been simulating, and somebody has an input on these.
Yeah, I think it may be a good transition from biological to moving into the philosophical or real human experience-based science. Wonderful. Thank you.
Comment on this, that the brain simulation, I think one experiment is already done in 2015 by Kobe Research Laboratory in Japan, and they did experiment, simulated the whole brain activity for 1 second, and that 1 second 1 microsecond, 1 microsecond simulation they took with the largest supercomputer in the world in 2015. It took 30 minutes to gather the data, and the data was about 100 petabytes. So that humongous scale of data and humongous scale of simulation is already done. So they are doing, maybe they are in the future, they are going to do some more work on that.
Yeah, so Barbara, we already have a microsecond long dream.
Yeah. Okay,
Yeah.
so Dr. Barsegov, I was just having some additional questions about this. You only simulated the kinetic core movement end on, right? There are many movements like lateral movements. There is an angular movement also. So those are not included in your simulations, right?
No, we have, with regards to the to chromosomes and the poles and microtubules, we have all kinds of things. Everything is in place. Of course, cell is much more, there is much more complicated object. So, for example, you have actomyosin network, which controls the cell motility, and that will be when we merge two models, for example, for actomyosin network and for microtubule kinetochore dynamics. So right now we're only able to simulate the motion of chromosomes, but of course there are other elements that we can take into account. Whether these elements will affect the accuracy, will affect the outcome, is hard to know. I'm sure that they will, but to what extent, it's not well understood. But with regards to the motion of chromosomes, we have everything as realistic as possible in the model.
Okay, and there are some other parameter regarding only the movement of the kinetic core and the chromosome. It requires, like we have a different kinds of vibration, stretching, non-stretching, and there are 4 to 5. Yeah. So, but in the kinetochore movement also, the chromosome movement, there is a monotelic, syntelic, amphiteric, these kind of movements are also there, where the tension between two chromosomes are different during the mitosis process.
Yes, we have the tension, we have the mechanics there. So once we have chromosome arms, we know how they're going away from one another, coming closer. We have the mechanics of this motion too. So we have all the stretching, all the bending in there, and then the kinetics of attachments. Now we have three motors, and we have realistic representation of the chromosomes themselves. We have cylinders, and so you saw these these big arms. We're good to go to start describing 10 or 20 chromosomes. So we're adding little by little different elements into the model, and as we do this, we're able to clarify certain questions that nobody knew the answer for. We have 2 papers published already, and we're working on the 3rd. One thing is that it's the rates of detachment are actually force dependent, and I can tell you one. I can give you heads up, and that is, if you model that, so you don't assume that the rate constant is constant, but it's actually a function of force, and it has been measured for different molecular motors, in the single molecule experiments, and the experimentalists have shown that these rates of detachments are actually force dependent. Once we model that, we have much better improvement in terms of the statistics of proper attachments. Okay. So we're getting there. So we have already implemented some of the mechanics elements in the model, and there's still more to be added to the model. And as we add one more new element into the model, the improvement increases, the improvement grows.
Okay. And is it possible to use this model for screening a drug, or checking whether the drug is binding to either microtubule or the different parts of the chromosome? Can we simulate something like that?
I mean, if you are exploring it, if you're trying to understand how the mechanism, what is the mechanism, then you really have to implement full atomic or even quantum simulations. But if you already know the rate, and you want to build a more complex picture with all these rates, and diffusion, etc., then you select this level of detail, this level of description. You can model the effect. You can model the effect of different drugs on different drugs in the cell. Yes. So it really depends on what questions you want to address. Either you use the all atomic.
I discussed with you about our cancer projects, and we are finding some good information into that one. So I was thinking that if we can just expose your system with those molecules and then see what kind of effect is there on the because cancer is always related to mutation of the chromosome. So if that is possible, then maybe we will discuss this later. But that's what I thought actually.
Yes. I mean, we can talk about it. Yes. See what we can do. Yeah.
Thank you, Dr. Valeri. If anybody has any other questions, so we can take one more question.
Yeah. I have one question. Actually, different cells, like different pathological cells, consider cancer cells divide faster than normal cells, and other cells like some neuronal cell or stem cell divide faster. So in that case, the kinetics of kinetochore and microtubule assembly is faster. So can we apply this model, because their error rate is high in comparison to this one? So can we apply this model in the cancer and other cells?
Yes, we can. Yes. The answer is yes. So if we implement the rates, if you change the rates and use the rates that are observed for the cancer cells, then we will basically describe the process of prometaphase in the cancer cells. Yes.
Okay. Thank you.
And yes, I also have I actually have two questions. Am I audible?
Yeah,
sorry,
please
but
go ahead.
yes,
Yeah.
but I only have time for one question. I really have to go.
Okay. So I just want to ask, like, my question is also on the application. Like Avindana, I want to ask about the chromosomal aberration that is there in the cell sometimes. So and this is the simulation study. So can we, if it is any way possible, to actually find out that if there is something gone wrong with the actual movement of kinetochore when chromosomal aberrations are happening using this model?
Yes, the answer is yes. We can try addressing different questions. These are associated with error. There's a thing called error correction mechanisms, and there are lots of these different mechanisms for correcting the errors. So yes, this platform can be used to explore those mechanisms. Yes, and we can use that. We can use this to address these questions. Yes.
Okay, and just a short question. You mentioned about the NDC80. So why just NDC80 linkers?
Well, because these are the main microtubule associated proteins. There are more, of course, and depending on the cells, there are different types of these linkers. We decided to implement just one. We can have more than one. We can have we can have two, for example, with their rates of attachment detachment, with their stiffness, etc. So we can add as many as we want. Yes, this model is amenable. This model is amenable to extension. Yes.
Okay, so that could be a future work, right?
Yep, one of the future directions. Yeah.
Okay, so now we had to conclude this. Thank you, Dr. Valeri, for this wonderful talk, and you are very closely associated with our INADS team, so it would be pleasure for us to hear you again for further developments. Yeah.
Thanks for inviting.
It's our pleasure. So we are concluding here this KSAS talk series, and we will come with new speaker in next month with some great ideas. So, and we will inform you through our email and WhatsApps. So thank you very much. Thank you all. Thanks.
Thank you, Dr. Barsegov.
Thank you, Raj. Thank you, Balaram. Thank you all. Many thanks to everybody.
Yeah, everyone.
Bye-bye.
Thank you.
Record.